So welcome everybody. So we started these, I think this is a little too loud. We have to use the mic because my voice isn't loud enough apparently. So we started having these faculty chats this year, and it's a great way to learn a little bit more firsthand of what's going on in different labs, and for you to students to have some time to really learn the state of the art, and also do some direct Q&A with faculty. So today's topic is engineering in the brain, essentially, and what we're doing at the interface with neuroscience. So we have two of our really renowned faculty in this area. I'm going to give a little background on each, and then we'll hear from each and start having our conversation. So we have Elizabeth Hillman, who's a professor in biomedical engineering and also a professor in the Department of Radiology, and an investigator at the Zuckerman Mind-Brain Behavior Institute, which is located on our new campus in Manhattanville. Professor Hillman joined the faculty of Columbia in 2006. She holds a BS and MS in physics and a PhD in medical physics and bioengineering from University College London, and she earned that in 2002. So before coming to Columbia in 2006, she held various positions, scientific positions and postdoctoral positions at Harvard and Mass General Hospital and Harvard Medical School and Mass General's Hospital Department of Radiology. So we're going to hear more directly on Elizabeth's research, and so I'm not going to say too much about that, but I think you'll find it absolutely fascinating if you don't know about it already. She's also a fellow of the American Institute for Medical and Biological Engineering, the Society of Photonics in Industry and Engineering, and the Optical Society of America. She has earned a number of other awards: the NSF Career Award, the Adolph Lohm Medal from the Optical Society of America, the Rodriguez Jr. Faculty Award from Columbia, and a Coulter Foundation Early Career Award. We will also hear from Alyssa Canafagua, who's the Robert and Margaret Hariri Professor of Biomedical Engineering and also a Professor of Radiology. She also currently chairs our graduate program in Biomedical Engineering. She joined the faculty here at Columbia in 2003. Her BS is in chemical physics from Université de Paris. Do I say that correctly?
Yes, very good.
Okay.
Excellent.
MS in Biomedical Engineering from Imperial College in London, and a PhD in Biomedical Engineering from the University of Houston in 1999. So prior to joining Columbia, she was a postdoctoral fellow and instructor at Brigham and Women's Hospital and Harvard Medical School. So Alyssa is a fellow of the Acoustical Society of America and the American Institute for Medical and Biological Engineering. Her other awards and honors include an NSF Career Award, the Bodasaki Foundation Award of Scientific Excellence in Applied Science. Did I say that correctly?
Yes,
oh,
very
goodness,
good. Very good. It's a mouthful.
This one, the Nagai.
Yes.
The Nagai Award of the National Institute of Biomedical Imaging and Bioengineering, a new investigator award from the American Institute of Ultrasound and Medicine, and a Young Investigator Award from the Acoustical Society of America. So I think what we'll hear is a bit of how they're really bringing some really novel physics and engineering to help us understand the brain, as well as other biological phenomena. And so I'm going to let them speak on that themselves, and then we can start a conversation. So Elizabeth, we will start with you, and I think you have some slides.
Yep. Okay. Can you hear me? Physics. Okay, so it's very unusual for me to only have 4 slides, so this is going to hurt. Okay, so what do I do? I use optics and light and lasers to image living tissue. That's been the theme of my research since I was a graduate student. And so you can look at all sorts of interactions between light and tissue, from looking at the absorption of hemoglobin to looking at fluorescence. And what's really amazing right now in research is that we have this ability to put fluorophores into living tissues, into living animals. So you don't necessarily do this in humans, but it means that in a mouse or in a fly or a worm or any kind of model organism, you can do some really, really creative things to actually make cells be very specifically labeled or make them actually show their activity by flashing and so on. And so I focused a lot on trying essentially to develop tools that researchers can use to be able to get this kind of information out of these animal living model systems. And so what you're seeing here is activity in the brain of a mouse using a technique that I developed called scape microscopy, which allows us to do really, really high-speed 3D microscopy. And this has been a big problem in the field for a long time. We've now got these beautiful probes that flash and do stuff in the tissues, but we only really had the capability to image them in a single plane, which meant that what we could learn about the system in 3 dimensions was really very limited. And so we came up with this method. It's a crazy, weird way of doing it where you create like a sheet of light into the tissue instead of scanning a spot around like you do in confocal or 2-photon. And then you actually move the sheet backwards and forwards to create a 3-dimensional image. And it's quite odd as well because it's actually it's faster and simpler and cheaper than a lot of other technologies. And I couldn't even begin to explain how we came up with it. It was just a whole big mashup of different ideas, and then we kind of just kept pushing on it and discovered that it was actually really, really effective. And so we took this to a company called Leica Microsystems, one of the biggest microscope companies in the world, and they have licensed it, and so they're developing a commercial product, and then we're kind of like now going crazy trying to help people use it. And another part of it is really to try to understand what we can use it for. And so although we can image things in 3D really fast, we can now do things like image things that move. And so this is a fruit fly larva that's crawling along, and we're tracking its cells along its body wall, and we're looking at how those cells move as it crawls, but also how those cells are firing to tell the larva where its body is. And so this kind of experiment was just unprecedented before we had this imaging capability. And then, as I said, we're extending this in all different directions. So there's huge amounts of applications in neuroscience, but this is an example of imaging a beating heart of a zebrafish, where we can see every single red blood cell going through the heart beating in 3 dimensions. And again, this is just something that we couldn't do before. Okay, I'm going on too long, I guess. But so that's one area of my lab is this microscope where we're really now just going nuts trying to develop it in every different direction and find collaborators to start using it and help people all over the world to build these systems for themselves. The other area that we're really excited about, we call wide field optical mapping, and it's again using these mice that have these green fluorescent proteins in their brain. And so their neurons actually flash when the neurons fire. And although we were trying to image them on a cellular level, we discovered that we could image them on kind of a bulk level as well. And that's super simple. You just use LEDs. When you shine a blue LED on, you see green fluorescence. And then when you shine a green or a red LED on, you can see reflectance, which tells you about hemoglobin. And so I use this in my lab actually to study the way that the brain controls blood flow. And that's really important because human functional magnetic resonance imaging that you may have heard of actually measures a signal that's derived from a change in blood flow. So initially when I got into this, we were trying to study the relationship between neural activity and blood flow. But as we were doing this, we prepared this really cool method that let us actually look at neural activity across the whole scale of the mouse brain. And so we can create these animals where we can image them while they're awake, we can image them over a long time, we can teach them things, and as they're doing all of this stuff, we can really see in real time the activity that's going on in their brain. And so we're using this to learn all kinds of stuff. So this is an example of data just when I flick the whisker. The image on the left is showing the neural response, which is a flash in the whisker region, and then the other ones are showing oxy, deoxy, and total hemoglobin, which are showing you the blood flow changes that go along with that neural activity. And so we've used this in my lab to answer a lot of biological questions. I think as a physicist, I like to figure out how things work. So getting into like the mechanisms of how a cell talks to another cell is really attractive to me, as well as tubes and pipes and flow and stuff. And so we actually discovered that the endothelial cells of the blood vessels are really important as a conductive conduit for propagating this dilation signal that happens when neurons fire in the brain. And that's given us this really cool insight into the way that brain diseases might actually be related to cardiovascular diseases in a different way than we thought before, which now we're using to explore different drugs that might be able to treat those diseases. We've done a lot of work looking at development and the development of this system, because the newborn baby brain actually has different fMRI responses to the adult brain, and so we did some experiments in mice to sort of look at exactly how that progression occurs, and we found that it does actually develop during brain development, which has a lot of implications for the care of premature babies, things like that. And then most recently, we just got this really wacky result where we were able to capture this sort of real-time ongoing activity in the brain, which we think is the neural signal behind resting state functional magnetic resonance imaging, but which also, again, gives us this kind of depiction of really what might be happening in your brain right now as you're sitting there. And it was just something that nobody thought was happening before. And I did a talk yesterday where I said, you know, when you do imaging, you really don't have a choice what you discover. Like you see something, and then it's on you to figure out how to explain it, right? And so we've had a lot of fun trying to understand this, and we just got a big grant from the Brain Initiative to dig into this as well and understand what that's all about. And that's it.
Okay, all right.
It was short for you.
It was, right?
All right, so hi everyone. I guess I'm gonna go to the next slide. Maybe
Oh,
not.
schizophrenia, yeah.
You want to say something about that?
No.
All right, so we use ultrasound, so different waves from what Elizabeth talked about, slower waves, and most of you may be familiar with ultrasound from the imaging point of view. What I'm going to talk about today is more about the therapeutics of ultrasound. So I don't have exactly a physics slide to show you, but this is an illustration about how we can have the ultrasound beam that you see vertically here activate some microbubbles that are actually flowing inside blood vessels in the brain. And then what happens, what you see there oscillating are the tight junctions between the bricks of the endothelial cells, which are basically, the bricks are aligning the inside of the vessels. And then they're actually set to open and vibrate according to what the microbubble is oscillating, the same kind of force. And then you can see the permeation of the drug molecules, which are in purple, to finally reach the actual neurons. So the activation of these vessels also affects the drugs that are actually going to reach the neurons, and that's actually called the blood-brain barrier. So it's exactly what it says. It's a barrier between what flows in the blood and what's actually reaching the tissue. And the rest of our body is very much has permeable vessels. Our brain is very much closed, and it has a lot of different cells that, other than the endothelial cells, that lead into more filtering of what flows in the circulation. So with ultrasound, we're trying to open temporarily the barrier and have the drugs reach the neurons, and especially for diseases such as Alzheimer's and Parkinson's that are affecting and escalating in the brain, leading overall to neuronal degeneration and shrinking of the brain overall. So what we do in my lab is a lot of what I just showed you in this illustration, but mainly also to develop, optimize these ultrasound beams. We also make our own bubbles. So these are micro bubbles, so micron-sized bubbles that are used as contrast agents for ultrasound imaging. So right now, if you had an ultrasound of the heart, and the cardiologist could not see your heart properly, they will inject in your radial vein, so these microbubbles that are basically resonators inside the brain, and they're used to reflect the ultrasound wave back to the sensor that's used for imaging, and to enhance the contrast of the underlying image. We actually use them to open the vessels, as I mentioned to you. And what you see there on the bottom left is the MRI, 3D MRI with two different regions in yellow that are showing where the gadolinium, which is a contrast in MRI, that normally does not permeate the blood-brain barrier, but it has in the regions where we focus the ultrasound. And then we use MRI to see where we opened. And then what you see in the middle panel are the neurons in brown staining that show you the region where we actually were able to restore the neuron in Parkinson's disease model. It's a toxin model called NTP. And on the other side, the contralateral side, the bottom picture in the middle panel shows you what happened to the neurons without treating them with ultrasound, and the drug called the Nutrien, which is a neurotrophic factor, helps the neuron regenerate after it permeates the blood-brain barrier. The one on the right is a video playing is of a mouse that looks like it's lifting its paw. So this we have the transducer that you can see, sorry, on the top of its head, and we use ultrasound without bubbles in this case to modulate the brain response that Elizabeth showed you, and that leads to a change in the motor response. One of the motor responses in the hind paw, and you can see that that can be just on the contralateral side, to one side of the mouse that can move. This is very counterintuitive, and because you are using mechanical waves now to electrically excite the brain, and then have a motor response. And on top, what you see there is something that we developed in real time, where you can actually monitor the response of these resonators, and you can see what the bubbles for the Bladder-Berry opening are doing. And the blue curve shows you that they actually stably resonate, which is something that you can see through the intact skull transcranially. And this is my last slide. I want to show you basically what happens at the cellular level, and maybe close the loop with what Elizabeth also showed you. So we have a neuron that looks that has this, this axons that it looks like it has tentacles. That's how neurons are. And it's labeled in green because it has expressed the green fluorescent protein that is after a virus, an adeno-associated virus that is used actually for gene delivery, has actually been expressed in that neuron. And you can see that only in the region where we have opened the actual barrier. And on the right, we see a panel where this co-localizes with neurons alone. And the cells that you see in purple and blue around it are the cells such as astrocytes or other cells in the brain that are mainly spared because of a promoter. So you can engineer the virus to be able to express the GFP only in neurons themselves at 95% rate. So the idea is to do targeted delivery in the neurons alone and help the cells restore and hopefully regenerate and stop the disease from progressing. I'll stop there, and the next is,
Okay.
yes.
Let me first thank you.
Thank you.
So maybe first let's talk a little bit about all the imaging, because it seems like the imaging is enabling us to really understand the brain, and then some of the imaging is actually translating into a therapeutic treatment, which is really fascinating. And so if we can talk a little bit, you both talked about the neuron activity and also the blood flow, and the ability to be able to look and understand both. Maybe if you can comment a little on how is that connected from a novice like myself, who might not know a lot about that, and how seeing that really can help us understand things.
Right. So, I mean, as I mentioned, like functional magnetic resonance imaging. Who here knows what functional magnetic resonance imaging is? fMRI. No? Who's heard of an MRI? Okay, better.
The F part.
Okay, good. Okay, so when you take an MRI scan, you're measuring these little protons, right? And they get affected by the presence of deoxyhemoglobin, which is a paramagnetic molecule that changes the way that they relax back down. And so when they were first doing MRI scans, they figured out that if they took lots of different scans of a person's brain, that they could actually see that something was changing. And so they did some sort of basic experiments, and they discovered it was because this concentration of deoxyhemoglobin was changing. So they named this signal the blood oxygen level dependent signal, BOLD, and they figured out lots of ways where you could use this to figure out what was going on in the brain. And you can have an X-ray of your brain, a CT scan of your brain, an ultrasound of your brain. It's not going to show you where the activity is. And so fMRI gave us this very first view of, you could just stick someone in a scanner, show them a bunch of pictures of kittens, and see which part of their brain was lighting up when they looked at a picture of a kitten. And we say lighting up because it was literally there was just like a bit of a signal change, and then you would like lock in on that signal change and say like that region right there is what's changing. And so that's given us this amazing view of the human brain and how it works. The way we used to do it was that people who had been in horrible accidents where like they'd had injuries where a piece of their brain had been damaged, and then they were like, oh, now you can't move your right hand. Okay, so now we know that that's the part that controls your right hand, and that's really how we had it before we had fMRI. So now we have fMRI, and it's used very widely, but because this signal is this kind of weird deoxyhemoglobin thing, it's very slow. And a lot of people even are confused by this name, BOLD, blood oxygen level dependent. People think it's like neurons are using oxygen and it's a sort of metabolic signal. But we needed to go in and look really carefully and say what exactly is happening. And what we confirm when we go and look really closely using our microscopes is that when the neurons fire, the blood vessels dilate. And they bring a whole bunch of fresh blood in, oxygenated blood, which actually decreases the concentration of deoxyhemoglobin. And that's what leads to the signal that you measure in the fMRI, BOLD signal. But it's like the vessels are responding to the presence of the neural activity, so it's a bit more slow, and it's sort of spread out because the vessels are really only operating in their vascular tree. And so we've got that far, and the neuroscientists want the neurons, they want high speed because, it takes about 3 seconds for this response to peak, whereas, by the time I'm making a movement, that's way too slow to represent what we normally do. And then in my work, as we sort of said, okay, first we want to discover what this link is. How can you interpret the BOLD signal in relation to neural activity? We got more interested in kind of actually the cellular mechanisms that link the two. And we discovered that it's quite possible that those mechanisms are vulnerable in disease states and can get damaged and broken. And so now we're actually really excited because we think we might be able to leverage that knowledge to use fMRI to actually look for disease or to understand the way in which a disease is progressing. And in fact, people don't use fMRI at all right now to do a diagnosis or to understand disease. And you ask them why, and they go, oh, because the signal's all over the place and weird when I look in a diseased brain. And we're like, yes, that's really important. But until we get this really good high signal to noise, careful, controlled data in these animal models where we can really understand what does it mean if it shifts a little bit later or a little bit earlier, what is the consequence of that, then we can't really use those methods in humans. So we do all of our work in mice, but we're not too far away now from being able to go test those hypotheses in humans using fMRI. It's a long answer.
So
sorry.
maybe before we get to therapy, you both work with mouse models. Do you want to first explain, since some people didn't know what fMRI was, maybe they might not know what mouse models were. Maybe you can explain a mouse model.
Wow, I'm no expert in mouse models, but so the model is a model of disease. So basically, and it's exactly what it says, the stress is on the model. So we're trying to, for example, have some of the phenotypes that are seen, or sometimes genotypes, so that means, the genes that are actually coding for the disease, and the manifestation of disease on the brain itself, right? So, and usually it's a model of parts of the disease in the human case, right? So for example, Alzheimer's has amyloid, has these proteins that are accumulating in the brain, and they're abnormal. They're not supposed to be there in the brain, and we don't really know exactly what triggers it. But when they do have accumulation of these proteins, then that triggers neuronal degeneration. So there's a lot of models for Alzheimer's disease, mouse models. I think there are about at least 40 at this point. And each one of them has the advantages and disadvantages. The way that you actually can do that is you can breed mice. So you basically have this gene that is exactly, perpetuated through mating in mice, and you have to, sometimes the gene has to be on both of these alleles on the DNA, or sometimes not. So sometimes you find yourself, breeding 40 animals only to find one has expressing, or two are expressing it. Others have a better yield rate, so it can be half of the mice having.
But now once you have,
so that's a
so
genetic
you
model.
can have these mouse models.
Yes.
So then when you can then image and see different types of response depending on the disease
and
state. I'm asking.
I can complete the mouse model because I didn't say the whole thing. So you can also induce disease without genotyping or genetically. You can also, for example, in Parkinson's mice, I mentioned the MPTP model. So that's a toxin that breaks down the neurons within a week. So it wipes out the dopaminergic neurons. So those are the ones that are firing and make sure that you can move legs and arms. And that can be the advantage of that is that it's much less expensive because you don't have to breed a lot of mice. You can just buy the toxin, inject to the mice, and then within 6 days you have the effect. But of course, it's controversial because that's not what happens in humans.
Right.
Plus, in humans, we're not necessarily genetically encoded. We don't know that whole story yet to have these diseases, be induced at that particular age in time as we do in
But
mice.
when you then look at mice that have these different, say, disease states, do you see very different brain response? And
Yeah.
Okay.
Yeah.
So can you
I
give
mean-
us an example of what we might see? I mean, I don't
Well,
know.
I
What
mean,
do we see?
A lot of neuroscientists actually study the normal brain. So in that
Right.
case, you can study just a regular mouse without worrying too much. But people still worry that, like, well, it's a mouse, it's not a person, and there have to be differences, right? And so some people who really, really get sort of anxious about that, then they work in humans, or the intermediate step
Right.
is to do monkeys. But it's funny. I remember we used to do rats, and everybody said, well, you can't teach a mouse to do anything. You have to do rats because you can teach them how to do things, and then you can look at, how they move their legs or how they recognize pictures and things like this. But, now people have figured out how to train mice. And so it's simply that you can use a lot of them. I mean, we're always really careful with them. We're always really respectful of them. We're not allowed to have them be in pain, anything like that. But you can do many, many things with them on a reasonably, short time scale that you just could not possibly do if you did it in a human, right? You don't have any control over it. So yes, sometimes we use just normal plain mice. I mean, just even understanding the normal brain is really hard, and we don't yet.
We go
For
to the
us,
subway and we get some. And then
we buy them. They're very carefully controlled.
Of course, no.
And we get weird ones that have glowing stuff in their brains and stuff as well. And then, but for example, we do stroke. So we can actually use a laser to create a clot in a blood vessel in the brain, and then we can immediately look exactly what happens. So you can see the blood flow stops, and then you can see the neurons just go crazy and start doing these like waves of activity, and then you see that affects the activity elsewhere in the brain, and you can track that over time and understand what's going on. And so there we've discovered stuff which then we go talk to stroke doctors, and they go, oh my gosh, that's such an interesting thing, because when somebody has a stroke, usually it's on one side of their body, but they have both sides affected to begin with, and then it kind of goes back to being on one side. But we can watch that in a mouse from the
Right.
very beginning all the way through their recovery, and you just can't do that in a human. So we start from mice. We get a decent idea of what we might think we might find, and then we have to then translate it more into like the human to make sure that we're not just studying something that's completely unique to mice.
And you each have very, cross-disciplinary collaborations, right? Elizabeth's just moved into the Mind-Brain Behavior Institute, where, and for closer collaborations with neuroscientists. Alyssa's lab is up at the medical campus right there with clinicians. Can you comment on how do those kinds of interactions influence your work?
I mean, I collaborate with both, and
Yes,
I think
right.
Elizabeth as well. So
Right.
they're completely different
Right.
ideas,
So,
and
yeah.
I mean, clinicians have the urgency of treating the brain and diagnosing it as well properly. So one of the, all these diseases that we talked about are very undertreated, severely undertreated, if not untreatable right now. So there's a big need. One part of the reason why I'm in the brain, I actually didn't do the brain before I came to Columbia, was because neurologists came to me and they said, we need to treat the brain. We need to have all kinds of tools there available for us. And I always say, if people are looking for ultrasound in the brain, that they're desperate, because they go MRI, PET, optical, CT, X-ray, and then ultrasound's on it. And so there's a lot of desperation on clinician side. I mean, Alzheimer's, Parkinson's, ALS, you name it, brain tumors. I don't want to exclude any disease here. They're just galloping forward. And one of the problems with mouse models is that you can treat a mouse with Alzheimer's disease, And there's been clinical trials, one after the other over the years, and they have been, just nosediving after, I mean, the billions and trillions that are spent by pharma. I don't even want to go there. So the clinicians are also desperate. The neuroscientists, as Elizabeth said, are actually, interested in understanding the normal brain, how it works. Because it's a lot that we don't understand in the normal brain, a lot, right? The Brain Initiative at NIH, NSF, and all that is actually part of that. So there's a lot of funding right now pouring in to understand the normal brain. And actually, one of the Brain Initiative ideas right now, guidelines, is to treat the healthy human brain, or to understand the healthy human brain. And this is the bottleneck of a lot of us. Like, how do you actually go the healthy human brain and probe it and do all this kind of cool stuff without hurting it, without damaging it? So there's a lot of, we don't have the right tools right now, all the right tools that we want. So we have to, trade off penetration for resolution. We know a lot about the surface of the brain, not much about, the underlying brain, human brain. Everybody wants cellular resolution or molecular resolution. I mean, so the physics is not helping us. So we have to do some tricks as engineers to use the physics for us. So and so far, I think it has been working, but there's a lot more to do.
Right.
Yeah.
So there is this big brain initiative in the U.S. and beyond to really try to bring all scientists, engineers, clinicians to really address how do we better understand the brain, and when we look at the just the few slides that you were able to show. It seems like there's a lot that we're able to see now, though, to start to understand these mechanisms, and how maybe you can talk a little bit about how when you first observe something that we've never been able to see before, how does it drive, because it drives the next thing you want to see, right? We always want more, and it also uncovers and changes how we were thinking something worked.
You put it on your phone and you show everyone, and everyone goes, "Oh my God!" So.
Actually, there is a process in science, and are there PhD students here? How many are PhDs? Okay, we have many undergraduates, right? Undergrads? Okay. Masters? Okay,
good combination.
So you guys are probably all doing research, hopefully.
And if not, you should be.
And if not, please do, and please come and meet us after this. If you do try to say anything. But so I think the process is that you see something for the first time, nobody believes you. They think you're crazy, okay? Then a couple of years later, after you show more and more of the stuff, and here you go again and again, and didn't mention this last year, but here I am again. And then people start, testing into their own labs. And then after another 2 years, they start publishing it, and sometimes they don't even reference you. And it's okay, and they think it's their own idea. So this is the process. It's a bit depressing, I know, but I think what it can actually show you, what it hopefully shows you, is that you have to believe in yourself. I mean, obviously, this is sometimes very trite to say, but it's true, especially in science, because you have so many people. And I sometimes I'm the naysayer, I have to say. Sometimes I'm saying, oh, this cannot. There's no way this works. There's no way these people are right in the head. So you have to also check yourself doing that to other people. But I think it's important to believe in what you see, and sometimes you have to see it 100 times to believe it. But you shouldn't dwell on this forever, because somebody else can actually invent that and discover that later, a year later, and publish faster than you. So there's a lot in be perceptive about your work, be present about seeing all these images, see something more than what you see, because an image obviously is, I don't know if it's 10,000 words, but at least
it's more than
it
that.
means sometimes a million words, and you have to see an image over and over again to actually distill the information. Sometimes it takes months of the same image. You go back to it and you're like, oh, I didn't see that before.
And so
you stared at it 1 million times.
If an image is 10,000 words, what's a video where you're actually
a video is even worse. Forget it. Billion.
But I would say one thing it's forced me to do is to go outside of my comfort zone into new areas where you see something and you're like, I have no idea what this is. So you sort of Google it a bit and you're like, okay, so I don't think anyone else has seen this before.
Yeah.
And then, you know, you stare at it a while. And I remember the thing I said about the endothelial cells, you know, we'd watched this so many times, we could tell that this signal was somehow spreading along a vessel, you know? And I remember I went all the way down the corridor, I asked all of my colleagues, can signals spread along vessels? And they're like, oh, I don't know, you know, go away. And so I Googled, like, does a signal spread along a vessel? You know, and I found this paper popped up that had just been published, and I read it, and it said, yeah, yes, you know, these signals can spread along this vessel, and it was from a woman in Australia, and I just picked up the phone and I phoned her, and I was like, okay, I see this thing in the brain, like, what the heck? And she sort of sat me down and talked me through this whole field and introduced me to it. And me and my students sat and just like surveyed all these papers. And then, you know, and within a year, I was like an expert in endothelial cells in the brain, and I could draw those diagrams with all the pathways and stuff. And I showed up at this conference, and so we published this paper, which, yes, got rejected like 100 times because everyone said I was completely wrong.
Yeah.
And then eventually, you know, this whole field was like, where did you come from? But you showed this thing that, you know, we never saw. And but I had to do it. There was nobody else was going to do it. So I had to just roll up my sleeves and be like, okay, I have never looked into vascular physiology before. And then you look at other people's work and you're like, wow, if they had just sat down and taught themselves this, they would have noticed this, right? But they didn't. So I want to blow the horn of biomedical engineering here, because I came from physics, you came from chemical bio, but in biomedical engineering, we are these integrators of information, and we're not afraid to just be like, oh, heart valves, I don't know anything about heart valves, and sit down, and in 2 days, you know, you can know a lot about heart valves. And so, you know, we have to do that. We have to keep moving forward and discovering. And I think if you just say you do imaging and you just do imaging, that's no good. When you have this privilege of seeing these things for the first time, it's really your responsibility to then like follow up and take it to the right person, or to take it on yourself if it's something that you think that you can do.
And then so it is bringing all these different fields together enables some really creative. I think of it as the innovation at the interfaces, because
Yes.
you're really seeing things at different fields intersect. And then, you know, Alyssa, if you can talk briefly about, you know, we think of ultrasound as an imaging method.
Yes. That's how
but-
you should think about
No,
it.
but I think you've also now shown it to be therapeutic, right? That we can take it as a way to bring therapy to and enable point-wise drug delivery. And so if you can talk a little bit about that, and then how do you make that transition? Because that's another kind of pretty bold transition.
Yeah,
right?
so I can't claim I discovered therapeutic ultrasound, but because actually it's much older than
Right.
ultrasound imaging. Therapeutic ultrasound, I mean, people were playing with waves. There's a Nature paper on bees in 1905, you know, focusing, you know, ultrasound waves and knocking down bees.
That's
Sorry to
awesome.
bees, yes, and so
That's why
it's
there's
been
no
around.
bees.
I wouldn't call
no?
that therapy, but
Yeah, but I mean, you have to test them sometimes.
Depends if you don't like
Before you go to humans. No, I mean, we know, obviously, you know, how ultrasound
Right.
could, you know,
could be
treat and also
focused.
have, yes, and by effects. So how
Right.
mechanically, thermally, you can change things. And then ultrasound imaging came about in the 50s, actually late 40s, when they figured out how to do, you know, beamformer and all that stuff. And then obviously so it's funny how two technologies, one came 50 years earlier, and the other one came, you know, and then took over the ultrasound field, and then people actually started revisiting. And now, you know, it has been very slow. Therapeutic ultrasound is actually in the clinic now. You can treat the brain, you can treat essential tremors. So if somebody has tremors as part of the Parkinson's disease, you can, you know, normally, you know, you can put DBS, which is basically pacemaker in the brain, major surgery. You have to insert electrodes you know, all the way to where your midbrain is. And you have to permanently have a device that's facing your brain. But with ultrasound, you just go for 10 minutes, and you see people, it's guided by MRI, you see patients that have the tremors, and literally they ablate the brain in very small region, and the tremor stops. Wow. So we've come a long way. It took a century in that case. But we're understanding also between burning the brain and imaging the brain, there's a huge other range where you can navigate within and cause minor mechanical effects like we cause with the stimulation, or minor thermal effects that can actually cause hyperthermia, where you can actually facilitate chemotherapeutic drugs to go to the brain tumor or other tumors. And one is also immunotherapy, which is now booming in cancer therapy. And there's an immune response now with ultrasound. So you can trigger immune responses. And if you actually focus your ultrasound beam on the tumor, then the T cells will go migrate into the tumor and try to stop the tumor from metastasizing. So there's a lot of techniques, new techniques in therapeutic ultrasound. It's just that we have to make sure that it works in humans before, because a lot of things are happening in mice, rats, you name it. But it's a very booming time for therapeutic ultrasound. For sure.
So we're going to open the floor for questions. Anyone have some questions? Yes. We have a microphone for you.
Thank you. You mentioned a lot about the technical challenges, and especially about neurostimulation and neuromodulation. And I would like to know if you think that the most challenging tasks are changing ethics and changing mentalities, because today it's really hard for people to imagine that we could act directly on the brain, or if it's mostly the technique and the science that is just a challenge.
Wow. The ethics on the brain. I think this is rapidly changing. I think, and especially if you're talking about disease, I think people, put their ethics aside and try to make sure that the patient can be saved or can prolong their survival. Are you talking about the healthy human brain? Understanding the healthy human brain, is that the ethical question that you're asking, or?
Not necessarily healthy, but,
Mind control.
Oh, mind control. Oh. Stimulation of the brain.
You have to have gel on your head, though, so you'd know, right?
Well, water works too. So,
Swimming.
The idea. So that's a good question. So stimulation of the brain now it's still is being done by drugs. We all know that. So the question is whether a device can do that. Now, mind control. I don't know if we, we could be there yet. I don't know, but they're just trying to basically replace what drugs can do with a device, especially if you have a tolerance or some kind of a allergic reaction to drugs. I mean, something about neuropathic pain, for example, has been around. We talked about this, obviously. Elizabeth led an opioid challenge. So how do you actually have replacement of drugs with devices? I think people are much more, not familiar, but comfortable with drugs and not necessarily with devices. I think invasive devices in neuroscience, they embrace that because it allows you to pinpoint exactly, where the cells are, where the different circuits of the brain are, and they can modulate them. We like transcranial, we like non-invasive, so the idea of wearing, a small ultrasound transducer cap on your skull is something that we also in midst of developing. So I think trying to broaden your mind beyond drugs, and of course with a big ethical, I agree with you. So so whether it's drug or devices, you have to inform any subject of any side effects or any problems down the line that they may find. And FDA has that down like a charm. So if we ever get to humans, that will be.
Anyone else have questions? Additional questions? Should I wait
I was
2
going to.
seconds? Go ahead.
I was thinking about one of the points that we made earlier about collaborations, and I wanted to say that, every year we have this Society for Neuroscience conference, which is like 30,000 people cramming into a poor city that has to cope with them. But it has by far the best trade show of any place that I ever go. And it's full of all these technology people, right? Imaging people, technology people, electrode people, everything else. And I always look at that to say that, the challenge of understanding and treating the human brain is one of the absolutely biggest things that we have right now. And it really, really takes a village. It's going to take input from all different fields. And one field that right now is a little bit, I think, lagging behind is data science. And so, you know, it both comes from, in our case, we have 1.5 terabytes an hour of data coming out of these things and their images. We have to turn that into knowledge. We have to turn that into signals, and then we have to understand like what it means. And this is an area I'm going crazy about right now, because we need this influx of people who understand these sort of deep learning models, networks, machine learning. All of those things are going to become absolutely essential to garner meaning from the kinds of data and images that we can currently get. So it spans everything from your technology, your biology, your geneticist, your chemist, your animal specialist, but now this need for data and information and analytics and modeling is blowing up in this field as well. And so I think that's something that's coming, and I think there's a lot of opportunities here
when you think about that, you've got especially with the imaging, that spatially you're able to see at smaller and smaller length scales, and then temporally you're able to see at shorter and longer time scales, right? So that's an explosion in how we're going to be able to be measuring.
Right.
But then we have to have the algorithms to really try to efficiently understand this.
And there's a big move. I mean, this has happened already in fMRI, where they've aggregated data from many, many places, the Human Connect Home project. And that's starting to happen in neuroscience. It's kind of clumsy right now, but you know, this idea that if you get more and more and more data, you can start to use these big data approaches to start to understand stuff. So that's going to, as I said, become a really big aspect.
And we also have some computer science faculty who are interested in looking at that in the inverse.
Mm-hmm.
If
Absolutely.
we can understand, you know, our brain is really an incredible computer. And if we can understand how our brain so efficiently stores and processes information, that it can guide how we should actually be designing computers. So because they're clearly not designed as efficiently as they could be, given all that our brain can actually
We've always
do.
been inspired by the brain. Neural
Yes,
networks have been
absolutely.
around forever,
Yes.
and that's basically where it
That's
comes
right,
from. So
right.
being able to access different information and be able to have clusters that are running all this pattern recognition, for example,
Yes.
and machine learning from being able to simulate what the brain does. And so the more we learn about the brain, the better we can engineer the computers to understand the brain. So it comes full circle.
Exactly.
So engineering is in all of
That's right.
in all of
That's
these
right.
different sources. So.
Yes, in the back. Oh, right here too. Sorry, couldn't see.
Hi, I would ask about diseases. We have a lot of neurodegenerative diseases like Parkinson's and others. And I was wondering if there's a possibility so when you're thinking about how to cure disease or how to make things better, is there a possibility of, say, fixing the brain, like the stroke, somehow finding a way to reconnect circuits that have died?
That's, I think, very hard. I think what you find is you can't really have a neuron survive after a while. So they reach a cycle in neurodegeneration where it's very hard for it to be reversible. So the idea right now in neurology is that you have to treat diseases early. And that's where imaging comes in, because we can't necessarily image at the early onset of Alzheimer's, Parkinson's, and all these diseases. So this is where imaging becomes very important, to be sensitive, to be accurate and precise, and detect whatever manifestation of disease at the early onset, so that you can treat it and make it reversible. But after a while, especially in the case of neurodegenerative diseases, it's a little bit controversial, but most people believe that it spreads. So, early Alzheimer's starts from the entorhinal cortex, or the hippocampus, short memory center of the brain, and then spreads to the rest of the brain. Parkinson's starts from the striatum, so more region, the motor center of the brain, and then it spreads from there. So if you have the entire brain already, being affected, it doesn't become just a Parkinson's disease anymore. It's affecting all the other centers in the brain. It's just much more difficult to treat. Stroke, I don't know. Stroke, if you can treat it at late stages, that is something I don't know. So.
I think the weird thing about, well, obviously the brain, it's very complicated. So we have plasticity in the brain, right? You have people who have had a piece of their brain damaged, and actually they can recover function, or the brain has ways of compensating. There have been children, young babies, who have had strokes where half their brain has been pretty much just destroyed, and they have normal function. There are some very, very strange things that we don't understand yet. And I think, I don't want to go too crazy here, but like, I think more and more we're also starting to understand why things that you wouldn't normally call normal medicine actually are effective for things like depression or even actual, brain disease states. Say, well, go and exercise more and you'll feel better. I mean, we're actually starting to understand that these interactions are biological interactions that are meaningful. So I think progress is going to be made in a lot of different ways, where we're going to understand better how, say, therapy in an autistic child might be actually remodeling the brain in some way and causing it to be improved, right? We're learning all of that now. But it's like trying to fix a car when that car is just so incredibly complex you can't even figure out how to open the door. It's yeah.
In the back a minute.
Yeah, I think you just started talking about what I was going to ask about. And so I
Microbiome.
guess I'll ask it anyway. Maybe we'll get a little bit more detail. And so my question is more in regards to like therapeutics when it comes to brain. And so, unlike any other organ, any therapy to the brain will cause some behavior change, or has a very high chance of causing some sort of behavior change. And so how do you see the role of like clinical psychology, or I guess psychology in general, playing more of like an integrative or collaborative role in this type of field?
I mean, I know a lot of people in psychiatry who are really interested in merging together this sort of their understanding of how you sort of treat a person holistically versus what imbalances might be driving the symptoms that they're observing. And I don't know what the answer is, but I think there's more and more people now who are studying, directly, like, you can take an antidepressant and it makes you not depressed, but actually some of them make you suicidal and all of these things. Like, we haven't studied that enough to really understand what it means to be taking those drugs, but that's starting to happen. The head of the National Institute of Mental Health now is a guy called Josh Gordon. He used to be a professor here at Columbia in psychiatry, and he was doing research in mice trying to understand which neurons in the brain regulate anxiety, while he was also a practicing psychiatrist treating people in New York with anxiety. There's a guy called Carl Diceroth who invented this technique called optogenetics, which is a little bit like the sort of therapeutic ultrasound. You can use light to turn neurons on and off in a mouse's brain, and he's a psychiatrist as well by training. And he got into this because he was like, I can't take it anymore, trying to use these different methods with these people and not really understanding how it all ties together. But it's coming. It's just it requires, as I said, again, this very, very integrated approach between trying to look at the cellular side, the chemical side, and the psychology side. Yeah.
But it's true that behavior is very important, because if you can change a cell and you can change, a vessel, but if it doesn't manifest in behavior, then people are very skeptical from the clinical side. So definitely behavior is a huge component of treatment.
Hi. So my questions for Professor Marcoglio. It's related to your neuromodulation project. So in terms of neuromodulation, it seems recent evidence showing that there's not one sort of stimulation that really provides therapeutic effects for people. It can vary person to person, and so labs can develop certain models to try and create a person-specific feedback loop. Are there any currently accepted models of feedback that we're using focused ultrasound for the brain that your lab uses, or is that something that your lab might be open to?
Well, we are limited by the imaging, so that's basically what we're talking about, the human level. So we modulate subcortical regions, and we want to understand, what kind of ion channels we potentially open, and what kind of mechanical transduction, so what kind of mechanical or electrical signals we are inducing. And there's, the only thing is fMRI right now, and as you heard, there's a lot of challenges. We're not doing this in my lab yet, but there are papers out there that have shown that ultrasound can stimulate fMRI responses and EEG responses as well. And I think the challenge there is more the motor response, because all other stimulation techniques have this magnificent response of being able to stimulate very easily in some subjects and not stimulating in others. So this is because we don't understand exactly how the brain stimulates. What are we doing when we stimulate with whatever, magnetic, electrical, ultrasound, you name it, or even optogenetics is different because it's more molecular. But we have to understand the brain to understand also why it's firing, why it's excited, why it's inhibited, and also the responses that come with it. So the answer is no, we don't have a good feedback.
So I think some of the last couple of questions were also getting on, maybe patient or individual specific. And you talked about how data science now needs to come in to help with understand and process and provide some really more complete information from all the imaging that we're able to do now and how that's going to grow. But another element is how we're having more and more genetic information about individuals, and that how some treatments work for one individual but don't work for another. And is that connected to some genetic makeup?
I mean, that's a super exciting field, and I think it's progressing in parallel with all of these imaging advances. I think they call it radiogenomics or something now, right?
Oh, yeah. Okay.
Trying to put together that information. There's one example, and I apologize if this turns out not to be, this is just an anecdote someone told me, but of a drug trial that was used to treat a particular kind of brain cancer. And they sort of didn't see a significant enough effect of this drug, so they abandoned it, but later on they went back and discovered that there was 2 different subtypes of cancer, and in the subtype, it cured all of them, right? Or there are stories like this where we start out, we don't really know what we're doing, and you said, there's all these drug trials for Alzheimer's, and you give them to a bunch of patients, and you just assume they're gonna work in everybody, and when they only work in 20%, you say, oh, well, this isn't worth it. But we're really interested in the point of time at which your disease is progressing. Certain drugs may only work in a certain therapeutic window, and when you have just patients enrolling in a trial, you've got no information whatsoever about where they are in their disease, and so you can't then interpret your results. And so absolutely, as we get this more and more information about genetics, about any additional information, these kinds of big data techniques as well can pull that together. And I think a lot of it is about sorting things out into the right piles and understanding then how to interpret what you get. We haven't had that before, and that makes me think we've thrown away a lot of potentially good options because statistics didn't take that stuff into account in the past.
Okay, one more question.
I had a question about the data science part. Can you give some more specific examples where data science can help with the data?
Sure. I mean, I have a video of all the activity going on in the brain of a fly while the fly is walking along on a little ball and turning left and turning right and everything else. And so far, we can stare at that data. But, and they have similar with the mice, the mice are we've got all these recordings of the mouse's brain, and every now and again the mouse suddenly starts to run and then stops and then starts to run again. So we're saying, well, okay, if I take that data and I develop a model, can I now, based on just the activity of the brain, tell whether that animal's about to run or not? Right? If you were going to do that just sort of in a basic way, you can average all the trials, but but you want to develop something more significant, which really points to which areas of the brain and at what point in time do they then predict when that animal's going to then run. And then you then have all these other variables happening as well, like it's whisking, and then there might be sounds in the room, and there might be the other things going on. And you know, if you're just trying to do that from a basic analytical approach, you know, you lose track real quick. But you can use big data analysis methods where you can just literally put, you know, all of the data and all of the covariates into the model, and then it's going to start to tell you, you know, what's important, what's not important, what factors are playing a role. And then you can backtrack that and go, okay, now I know I have to look at this area of the brain for a signal that represents starting to move, right? And in fact, those signals are really important for Parkinson's, right? And so, but you can't ever get that from the standard current way people do experiments in these animals, because everybody else, you just train them to do a task when you shine a light and then they do something. So you never get that anticipating, preemptive part of it. So now more and more in my lab, we're getting this crazy data, which is all this sort of spontaneous stuff. And so we're going to need those approaches for sure there. And that's just one example. I mean, we've got loads more. But.
We talked about it, that, you know, every video is 1 billion words or something like that. So, you know, our brain is a good computer, but not that good. So what did you begin? I mean, you see an image like that, what do? Do you do a frequency response? Do you look at the spectrum? Do you look at the intensity? Do you look at the phase? So there's so many ways that you can process the same image, right? So you have to have all these parameters and run them through some kind of a algorithm, and really fast. Obviously, that shouldn't take years. So that's the challenge about distilling in very important information that by eye, what we're doing right now, radiology, for example, which is basically the clinical version of imaging in the clinic, is all by eye. It's all basically, you know, clinicians that have been trained over 10,000 images. I call them pilots, because you have to have 10,000 hours of images that you train your brain. You basically do pattern recognition in your brain effectively, and then finally you can diagnose somebody, right? But can we do much more than that? Can we actually have also, you know, some type of a, you know, machine learning going at the same time, and then be able to do, you know, millions of imaging in hundreds of thousands of patients? They can train a network with it. Right now it's all ongoing.
They're doing it, yeah. They're starting to use it.
If you guys are interested in that, it's a huge field, machine learning and imaging. I mean, they're still trying to break through with an example, like you say, here's what and the radiologists didn't see. I mean, it's a challenge because you have to have engineers, computer engineers that understand physiology. And that's where biomedical engineering comes in, and you have to understand what you're looking at. If you just do this blind processing, then it's not as informed yet. So you have to inform your algorithm enough to be able to distill, to train your algorithm to be able to distill the right information. So that's a challenge.
Which is why it's so important to collaborate and to have like combined, you know, real immersion into the field as well as the data science skills.
Okay, so we have a couple more questions, and then I have something I want to close with. So.
Actually, I want to follow up on that data science questions. Because I'm working on deep learning, so this is like new training machine learning technique. But one problem for deep learning is you can't really understand how that the model works. Sometimes it works very well, like you feed the model a bunch of data, and maybe you can know this patient whether has this disease or not, but you don't know like why this patient, or like how does the model decide this patient has this disease or not. So I don't know if this will become a problem for you. Like you can really use this deep learning model to do things.
Well, it depends. I mean, in radiology, we talked to the executive vice chair for research in radiology recently, and he is using machine learning to basically just simply prioritize cases. Okay, so normally he says, you know, normally they just kind of read these X-rays in chronological order, but a first order sweep, just to say, you know, is it possible that the serious, you know, to try and find the serious cases without someone having to spend a lot of time, just to put them at the top of the list, right? So something like that, I don't think it's such a big deal. You're not depending on it. You're not trying to replace a radiologist, but you're using it in a way to prioritize. So I mean, something like that is pretty good. But I would say, okay, it seems like it's the next frontier of data science to not say that, right? There are methods where you can back out, you know,
Right.
what are the prominent components. There are methods where you can actually generate, you know, different kinds of data sets where you can start to probe and understand those models. I mean, Google doesn't care. Google wants to fill in the rest of your sentence for you, and it doesn't matter if it's wrong or right. But for this kind of thing, it really does, right? But we have the capability that it's not far off where that isn't going to be the answer.
Yeah. I'd
Yeah.
also say, because from someone who's in the area of materials, that I've been thinking about this kind of question a lot from a materials science perspective, because we're able to simulate and measure at smaller and smaller scales there. And then, so we're talking about, okay, now what if we throw data science algorithms at some enormous molecular simulation? And you're right, the data science algorithms, deep learning isn't finding the mechanism, but it might be finding where something's happening. And then
Where to look.
Then it points the more physics-based either imaging or modeling or experimentalists to go where you should be looking, over what time scale, over what length scale, because this is where something that's really fundamental happening. So I think there can be a really unique combination.
As long as the
you
two people
bring
talk
these-
to each other.
Exactly.
That is
That's
so
right,
important.
right. But I think that brings up how much engineering and science now are teamwork and medicine. That it's not, you don't just sit by yourself if you really want to make an impact. So one more question here.
I actually have two questions. The first one is a follow-up for the data science one. I have limited experience with dealing with deep learning and brain imaging before, and I tried the deep learning model on brain image, and it doesn't, I mean, just for each data, it doesn't really work in my case. And I found some really weird patterns I learned from those data, which doesn't make sense in the neuroscience view, or we cannot interpret those patterns using the current knowledge. So I'm curious about if you have any experience with, if you have similar experience, you find some pattern that makes sense for the classification, but it doesn't really make sense in the neuroscience field and how you deal with those things. And another problem is about, question is about the resolution of the new imaging technique you mentioned, because I had a little experience with EEG and real-time control, real-time display of those signals. And I'm quite curious to know if these new imaging techniques would be helpful for us to build a more delicate, more accurate model to interpret the brain activity, and to build a more delicate, to make other, like exoskeleton, those things to do more delicate tasks. Thank you.
So I can answer the second one first. I think that the weird patterns of wacky activity I showed at the end in the mouse brain like that, part of the grant that we got is to get data from clinicians who stick electrodes into the brains of humans for epilepsy treatment. So doctors uptown, if someone has epilepsy, they stick these electrodes all throughout the brain now, and they keep them for 10 days. They do video surveillance of them, and then they are looking for if there's any seizure activity, if they can try to kind of localize where the seizures are. But we can have access to that data to really look and see if we can start to understand these patterns that we're seeing. So by using the human data, where we can get a sort of conventional electrode recordings, down to the mouse where we can get both those kinds of recordings and the cellular level recordings and stick an electrode in, we can span all of those scales. And that's exactly one of the things we said we wanted to do, was to sort of explain, like, what have we been calling those patterns all along? Can we match that disconnect between the sort of representation that you can get on a human versus what we can learn in a small animal? Which brings me also back to the machine learning, because you've got 10 days of recordings of a human patient doing everything humans do, and we have, hundreds of electrodes throughout the brain recording this activity. There's so much crazy stuff there that I think we could learn. I mean, to your first question, you know, of course, I think anytime you just take an algorithm and you put data in and you get nonsense out, you know, that happens all the time. One of the biggest skills to learn is to recognize nonsense and to keep, you know, trying, keep, you know, chipping away at trying to make sense of it. And I can't say it enough, I'm going to say it again. You've got to be there in the room with the person who built the system that generated that data, right? And the person who stuck those electrodes on the person. You know, you have to know as much as possible about where that information is coming from. Because, you know, I've had people in my lab who have analyzed something and made a mistake and been like, oh, I discovered this new thing. And I sort of grab them as they're running out the door to tell people. And it's like, come back. Okay, sit down. No, it can't be right. Keep trying. There's a bug in your code. There's something wrong, right? So that, again, is our job, to just really keep checking and trying and thinking and trying to either, as I said, find the explanation, which, you know, in some cases you can find an explanation and it's awesome. And other times you have to know when to be like, this is garbage. I'm just gonna, you know, there's something wrong. I need to do this again.
So can I want to ask a closing question from each, and then we can have some, you know, time to chat outside. Right now, I mean, we've seen so much happening over the last, say, 5 or so years, and in the research and the understanding of the brain, and yet we also know we haven't scratched the surface. If you think about, you know, all that's happening right now, not just in your labs but everywhere, on really trying to tackle this challenge, can you give a sense of where do you think we'll be in, say, 5 or maybe 10, maybe go 10 years, so you can be really wacky in your thoughts on what might we be able to see better, what might we be able to image better, what might we be able to understand better, and, you know, where will that take us in terms of, one, understanding the brain, but also helping in therapy for some of the really pressing needs.
I think, I mean, I don't know, I'm a little bit, it's very difficult with the tools that we have right now. And, you know, there's a lot of development going on. And 5 to 10 years, I think, is short.
Okay.
I think there's a
go
lot.
further.
Yeah, further. So if you go further, I mean, understanding how genetic disease manifests, you know, across different scales, from molecular to cellular to tissue to the disease state, I think, and if you have to pull together with all the different imaging and genetics going on and behavioral, and, you know, from models of disease all the way to humans. So we can have a full picture of what's happening, but you know, we don't have the tools right now. I think that's where I think engineering comes in, to get the right, you know, imaging tools, the right, you know, molecular probes, or the right, you know, data science algorithms. There's a lot of challenges that are happening within the different disciplines that need to come together. But the idea is to be able to have a full global imaging of the brain on all kinds, you know, and then tie in the different things, you know, the different aspects, from metabolism to electrical excitation to vascular flow. I mean, at different levels, because you have tiny vessels to big vessels, or neurons and all kinds of other cells. I mean, the downside, I think, of neuroscience is that they focus a lot on the neuron. I mean, that's only part of the brain.
Yeah.
There's so many other things going on at the same time. Yes, the neurons are important, and the neurons are firing, and that's very exciting. But there's so many other cells that we really know so little about. So there's an atrophic science going on, you know, neurocentric, hence the name. So, but I think in the end, we're going to understand how the cells come together, communicate, and make, you know, this information, you know, propagation that happens to, you know, to the rest of the body, but also, you know, with other sensory signals and other stuff. So, but, you know, I think we're going to be able to understand it, but I think it's going to take a century. I think.
So
it's going to take a lot of time.
I'm more optimistic, although anytime anyone says they want to understand the brain, I'm like, don't say that. That's such a vague statement, right? But I think, well, certainly the BRAIN initiative over the last 5 years has really, really accelerated things. And I was a skeptic in the beginning, but now it's like, we used to just wring our hands of like, oh, if only I had this thing. And now it's like the number of tools and the things that are available are incredible. And they're at the fingertips of so many more people. And at the fingertips now of people who do disease research, which is a huge transition that I've seen that hasn't really quite been recognized yet, but it's a big deal. You can look at brain activity in Alzheimer's. It's very different than the sort of standard structural methods of before. But so I think it's difficult. You assume like one day someone's going to be like, hey, we cured cancer, right? And you couldn't say that cancer's been cured. But 5 years ago, 10 years ago, we didn't have even a fraction of the choices that people have right now for detecting cancer and for treating cancer. Lisa mentioned immunotherapy. It's a huge new thing, and it creeps up on you. You barely really recognize it. And I think this is happening in neuroscience. Like I said, recognizing things like microbiome and the importance of all these extra different things. It's incremental. You don't see some big announcement. Now we understand the brain. Like, that's never going to happen. But these little pieces piece together and start to make sense. And I recently did a review of a paper that somebody published on how you could, if you eat too much salt, it causes a problem in your gut, which then actually affects your blood vessels in your brain and actually causes cognitive deficit. And they showed this, and they showed if they reduce the salt, then the cognitive deficit goes away. And they did it all in mice, and it worked beautifully. And it's like, this is now a new explanation for like why you shouldn't eat McDonald's all the time. But these are little pieces of information that become part of our culture that we understand. And so those little pieces, I think, start to guide us towards better practices that are going to help us. You think it was a nonsense, but it's it was these things. They sound so silly, right? But actually,
But I
we've
think
known
that
for a
gets
long
back
time.
to the thing of some things that are sort of natural treatments or homeopathic treatments. May actually be some real physics behind this that we will come to
our
learn and
conclusion
understand.
of our vessel work is that diet and exercise are probably good for your brain.
It is, and they actually measured a lot of growth factors there. Even the barrier opens, and there's more growth factors reaching the neurons. So it's great to exercise. Don't eat at McDonald's. I agree with that. But the placebo effect is very interesting too. So why? So Parkinson's disease patients, for example, you tell them you're going to be treated. We have the drug for you, and this is actually where behavior comes in. And all of a sudden, they stop having the symptoms, and it could be candy. It could be McDonald's, and they stop. So what is that, the placebo effect?
We have to take that seriously.
We don't really understand it, and it's fascinating that we can trick our brain to think something, and then release whatever, dopamine or whatever other, substances that we have naturally that can overtake whatever responses our brain is under the disease state. So it's very exciting to actually see that the brain can actually, trick itself to healing itself. So if we can harness that, that's one of the fascinating aspects that makes some of the salt thing, a little bit skeptical, but it could be something there that we need to understand. So again.
Wonderful.
It's very
Does this
robust.
work for Shake Shack too? We'll end there. And let's thank Elizabeth and Alyssa.